Good morning, everybody. Great to see all of you back after the beautiful party we had last night. Today we're going to start the second and last day, and for the first time a full day as well, of the ISCCM. The first session will be on the tissue engineering, but before we start with that, I'd like to ask everybody in the breaks, please not only visit the posters, please do, but also have a look at the sponsor booths just between the coffee corner here and the coffee corners in the back. And that having said, I would like to welcome very much Amy Rowat from UCLA. She's going to be our keynote speaker of the morning. She did her PhD in Denmark, then moved west to Harvard, then even more west to UCLA, where she still is, and I'm going to not make any mistakes. She's now a professor and vice chair of the Integrative Biology and Physiology at the Marcy Rothman Presidential Chair in Food Studies. And she did a lot of wonderful things there, gathered lots of prizes, but the most interesting thing I think is that she founded Science and Food, combination of things I think we all like and love around here. And today she's going to tell us about scaffolds and soluble factors to support growth of cells in many different tunable ways. So I'm looking forward to that. Amy, floor is yours.
Thank you very much for the kind introduction. It's such an honor to be here today, and I'm thrilled to be able to share with you some of our latest advances in how we're harnessing cues in the cellular environment to drive more efficient processes for growing cultured meat. I'm a biophysicist by training, and so a central question that really drives my work is understanding how cells integrate various mechanical and soluble cues from their environment to regulate their behaviors, functional behaviors such as how quickly the cells might grow and divide, or how a cell might be able to squeeze and migrate through a very narrow gap. If we could fully understand how cells integrate these different environmental cues to regulate these behaviors, this could enable us to really control the deformability of a cancer cell and block metastasis and how disease spreads through the body, or engineer a perfectly textured, tender, nutritious steak, slaughter-free. So in my group, our projects span the spectrum of human and planetary health applications. And to give you a sense of what that looks like, we've spent quite some efforts developing technologies to be able to quantify mechanical properties of cells, especially in a higher throughput fashion. We're working to understand how cells regulate these mechanical behaviors. And finally, we're towards the goals of human and planetary health. We're working to translate our findings for both developing more effective cancer treatments and also cultured meat. So in this field, a lot of times people ask, well, why did you get interested in food? A lot of our work has been focused on human health applications. But it turns out that food was really pivotal to my training as an experimental biophysicist. And later on in my career as a postdoc, I started seeing some of these connections between my science and food, and that sort of spawned the development of science and food, as Nika mentioned. As a postdoc at Harvard, I developed the first science and cooking class with colleagues and developed a similar class at UCLA, the science and food class. And these were all designed to use food and cooking as hands-on activities to engage students in learning science. Some of our interesting findings from our scientific bake-off can be found in this piece here, featured in the New York Times. And you can read about various other resources and curricula that we've developed over the years as well. And importantly, I think I'm very proud that we've hosted a series of public events over the years to engage thousands of people in critical evidence-based discussions around timely food issues. So you can find some of those pieces and videos on YouTube as well, if you're interested. But the central question that we're here to talk about today is our research on how we can grow in efficient, scalable ways cultured meat. So our vision really builds on the foundational work of pioneers in the field, such as Post. And our approach uses scaffolds to be able to support the growth of naturally adherent cells in suspension culture. So to give you an overview, some of the focus points of our work are using these mechanical and soluble cues to accelerate tissue growth, and then also developing methods to be able to build 3D tissues, such as the marbled cultured meat, which you heard in my graduate student Stephanie Kvesky's wonderful talk yesterday. So today I'm going to tell you a little bit more about the motivation from mechanobiology that really provides the basis for our work, and then our approaches using scaffolds to be able to culture both fat and muscle tissue, as well as some future projects that we're working on as well. So as we learned from some of the talks yesterday, these mechanical cues in the microenvironment can be very important for cultured meat on many different length scales. And while there's many efforts to develop methods to be able to grow cells without any scaffolds, scaffold-free culture, we do think that scaffolds can bring many advantages and benefits to cultured meat. We know that scaffolds could enhance the sensory and nutritional properties of cultured meat. Scaffolds also can provide structure across many different length scales, from the cellular level, providing guides for cells to align themselves, for example, to the tissue level scale in developing more structures and, for example, marbling in a multi-component tissue. The ability of scaffolds to introduce porosity can be important for facilitating the exchange of nutrients and waste products. And importantly, mechanical cues, as I've mentioned, can regulate some of these behaviors that are really important for cultured meat, such as how quickly cells can grow and how efficiently they might differentiate. So to give you a background of some of the key studies that have really motivated our work, this is a study from Penny Gilbert looking at the expansion of muscle stem cells, and she plated some of these muscle stem cells isolated from mice on more compliant PEG hydrogels. This is the modulus 12 kilopascals, that just is a measure of the stiffness of that substrate, and compared to cells that are plated on a stiff petri dish. And you can see over time the expansion of the cells on the softer, more compliant hydrogel is higher than the cells on the stiffer substrate. So that's expansion, very important for cultured meat production. Also differentiation, we know, can be driven by mechanical factors as well. This is some seminal work from Adam Engler in the lab of Dennis Disher, when he was in the lab of Dennis Disher, looking at the differentiation and myotube formation of CTC12 mouse myoblasts. And you can see on a substrate of about 11 kilopascal, the myotubes are very beautifully showing aligned nuclei and striations, suggesting that this is a more ideal substrate stiffness than a softer or stiffer matrix. So these are the muscle cells. We've also learned a lot about the importance of fat and adipose tissue engineering. And in this front, as Stephanie also mentioned yesterday, we know that the stiffness of the surrounding matrix can drive adipogenesis. In this paper here, we see that there's higher lipid accumulation for 3T3L1 cells embedded in alginate hydrogels that are more compliant compared to the stiffer gels. And similarly, you can see increased expression of PPR gamma, an adipogenic marker, for adipose-derived stem cells on softer rather than stiffer substrates. Again, supporting that the more compliant, softer matrices are important for driving adipogenesis. So in our group, we're using some of these insights from mechanobiology to be able to design customized edible scaffolds for the growth of both the muscle and the fat tissues. So we, of course, wanted to make our system compatible with suspension culture methods. So we took an approach of making edible microcarrier scaffolds that we could seed the cells onto and then culture in a spinner flask and then harvest. And this work was conducted by Sam as well as Stephanie, who you heard from yesterday. So the approach that we take to making our microcarriers is to use water and oil emulsions as a template. And we use gelatin as proof of concept, but this can be done with other polymers such as alginate, pectin as well. The polymers are cross-linked with microbial transglutaminase for extra stability, and then we collect them, break the emulsion, and we get these tiny hydrogel beads. This is what they look like, and we can also tune the stiffness, or Young's modulus, that we measure using atomic force microscopy, just by tuning the weight % of gelatin. So we also wanted to test the effect of topology of the microcarriers, because we also know from mechanobiology studies that aligned topologies can guide and promote the formation of myotubes. So we developed a process to make grooved microcarriers simply by templating the particles, squishing them between templates that we fabricate using soft lithography. And this created these grooved microcarriers that are like pancakes with grooves that are about 10 microns in width. We can tune the stiffness of these microcarriers as well, again, to match the optimal stiffnesses of matrices that have been found to be optimal for proliferation and differentiation. So the first question is, well, how do the cells interact with these microcarriers? And we can see both on the spherical and also the grooved microcarriers, comparing to cytodex microcarriers, that the cells are indeed adhering. You can see the nuclei labeled here in cyan, F-actin in white, and the beads are yellow. And we see over time that the number of cells tends to increase, which you can see by the increase in the number of cyan-colored dots or nuclei over time. And we see increased growth in all three situations. It's hard to tell exactly to quantify the cells given these confocal images. So we also conducted genomic DNA analysis to quantify the number of cells over time. And you can see that in all three cases, the cells are indeed growing on these beads in suspension culture, but we do see increased growth for the cells on our grooved microcarriers and the spherical microcarriers compared to the cytodex. So the cells, the microcarriers do seem to be supporting cell growth. We next asked about the differentiation potential, and this image is showing you some myotubes that have formed, labeled with the magenta myosin heavy chain, and you can see that in these 3D reconstructions here, these beautiful myotubes, the grooved microcarriers are shown here in yellow, and this is just one representative example. We found similar myotube formation for the spherical microcarriers as well, and we quantified the differentiation by quantitative image analysis and also by looking at the expression of myogenic markers as well by PCR analysis. So you can see that the myotubes notably are spanning hundreds of microns. In fact, they might be even longer, but the field of view limited us to this analysis of looking at these microtubes that are hundreds of microns. And we also analyzed assessed the volume of the microcarriers as well, which is between 30 to 40% or so, compared to the volume of the cells in these microtissues. Now, just for comparison, here's an image of some rat skeletal muscle, and you can see that these muscle fibers are very long and straight, so we're not quite replicating exactly the structure of the skeletal muscle, but we found that these structures that we're finding were more aligned and showed longer myotubes than for the cytodex bead situation. So you might have noticed that these cells and microcarriers are forming aggregates, and this is something that was very evident in culture with the C2C12 cells that we were using for proof of concept on the spherical and grooved microcarriers, also the cytodex beads, and this had been observed before, but because we're able to make the beads and the microcarriers out of edible materials, this enabled us to simply harvest these aggregates to form a meat kind of patty. Now, these samples, as I mentioned, were proof of concept with the mouse cells, and while there is a market in the pet food industry and other exotic foods for mouse meat, we also did want to show that bovine satellite muscle cells can be supported on these microcarriers as well. So thanks to the Kaplan lab for these bovine satellite muscle cells that also formed these aggregates with our microcarriers and could be formed into a patty that showed characteristics of myode reactions when in browning, such as the browning upon cooking. So this is an example of a very lean cultured meat product. But as we heard yesterday, the fat is very important. And so we next asked how, if our edible microcarriers could support the growth of adipocytes in culture. And there's Stephanie again, who you've already seen now. You've also seen this data that she showed yesterday in her talk, these beautiful images showing the cyan microcarriers here with both primary rabbit adipocytes and also mouse 3T3L1 cells growing on these microcarriers. After inducing differentiation, we can see that the lipids shown by yellow here in this lipid tox stain do accumulate over time, both for the rabbit and also for the mouse cells. So this shows us that these edible microcarriers are also supporting the formation of fat tissue that shows lipid accumulation. So the next big question is, well, how can we start to build marbled cultured meat and integrate the fat into the muscle component? You heard a brief overview of Stephanie's approach that she developed, stacking layers of the muscle and the fat microtissue. In this situation, Stephanie used electrospun scaffolds as a scaffold for the muscle cells, which she then differentiated to form this myogenic microtissue, and then took the beads seeded with the pre-adipocytes to generate the differentiated adipogenic microtissues, and then layered these together. So while you saw the overall outcome of this layering or marbling, I wanted to just give a bit more detail of this assembly process, which was very interesting for us to discover that these microtissues just spontaneously adhere to each other on the timescale of hours. And to assess that, we developed a very crude adhesion assay by just stacking the adipogenic microtissues on top of the myogenic microtissues. And they sit in this petri dish. You're looking at the top-down view, and we've outlined the microtissues with yellow. And what Stephanie did next was to nutate it, so mechanically perturb this stack of tissue. And what happens, you see, is that the adipogenic microtissue sort of falls off. It's displaced from the myogenic microtissue. And you can see this because the yellow now is showing you where the fat microtissue has been displaced to. So this is at 0 hours, so you wouldn't expect that there would be much attachment between the two different microtissue types. But over time, we can see that, that there's no displacement, very little displacement at 6 hours, even after 6 hours, or also 12 or 18. So this is showing us that the adipogenic microtissues are adhering, they're sticking to the myogenic microtissue. And we quantified this in terms of a relative adhesion index, building on this quantification of our image analysis data to show this, this relative adhesion index normalized to the median at time 0 for mouse cells on gelatin scaffolds. We also wondered, though, if this phenomenon could be seen across other species and other types of scaffolds. And so this data is showing you the same setup for our primary rabbit cells, the mouse, and also a scaffold-only situation on plant-based scaffolds. We worked with Matrix FT on this, who provided both Zane and soy scaffolds. So this is all showing you that, that this relative adhesion index increases over time, generally on the timescale of hours, for cells from different species and also different types of scaffolds. by contrast, the scaffold-only context here shows no change in relative adhesion index over time. So clearly the cells are somehow mediating the adhesion, the spontaneous adhesion of these microtissues to each other. So what does it really look like? Well, Stephanie showed also these beautiful cross-sections yesterday that she generated, showing, in this case, rabbit marbled meat, in contrast with the Wagyu beefsteak cross-section. And again, you can see, as she mentioned, that the length scale needs to still be tuned. The muscle here is shown in false colored in red, the fat component in yellow, but we are getting sort of relatively similar length scales, at least for the fat, and this process can easily be tuned. But I think the exciting part here was that these tissues are forming from the spontaneous adhesion, and they're forming these cohesive structures by the self-assembly. So we also did some mechanical analysis to understand, they look like the tissues, the microtissues are adhered to each other, but are they really sticking together? So we used rheometry in collaboration with Tom Mason to quantify the stress-strain response of both our cultured meat samples, also compared to the conventional Wagyu. And just to summarize here, you can see the Young's modulus that we've quantified. And you see that our microtissues, this is the multi-component microtissue MT here, has a lower Young's modulus than the Wagyu. But we were also interested to see that the multi-component tissue that we take directly out of culture has a lower Young's modulus as well. But we were able to tune and achieve an order of magnitude increase in this Young's modulus simply by dehydrating slightly the sample, just by wicking away the moisture overnight. So when it comes directly out of culture, it's clearly more compliant than the conventional Wagyu beef, which is also more dehydrated than and aged more than the multi-component tissue. So while we see overall qualitatively similar stress-strain response, the tissue is not falling apart. It's showing behavior of a solid, of a viscoelastic solid. We do see that we will be able to tune the texture, I think, by tuning the water content of these samples. So I think lots of interesting areas for study in the future. We also took a look at started to look at the nutritional and cooking properties of these multi-component tissues. This is assessing the raw protein content. We don't see any statistically significant differences, but the statistical power of these studies are limited because the samples were quite large and time-consuming to produce. So clearly more work to be done here, but it seems that we're in the right ballpark for protein content similar to the Wagyu beef. And looking at the cooking loss, we're seeing slightly increased cooking loss compared to the Wagyu samples when we've pan-fried these patties of the multi-component tissue that we've made. So again, no significant differences. We're sort of in the similar ballpark, and I think further tuning the water content into the future will could further reduce the cooking losses that we see as well. So to summarize, I've shown you how we've been able to use these edible microcarriers to be able to grow muscle and fat tissue in suspension culture. And we've been able to use the microtissues as building blocks to generate multi-component tissue, so towards marbled cultured meat. And we've begun to translate this approach to different species. Of course, there's more work to be done on this front, and we have experiments ongoing with bovine, porcine, ovine cells as well, on different types of scaffolds as well. So in just the remaining couple of minutes, I'll just spend a bit of time talking about some ongoing experiments that we're very excited about and directions of study. So I've told you largely today about the mechanical cues, how we're harnessing these mechanical cues and making customized scaffolds. We're also working to identify soluble cues that might accelerate desired phenotypes for cultured meat and fat as well. So this is one beautiful example in collaboration with Dr. Rochelle Crosby, who's here in our audience today. Thrilled that she was able to join us here. Rochelle's a muscle cell biologist by training with an extensive background and expertise in Duchenne muscular dystrophy, whose lab is next door to ours, and so we've had lots of interesting conversations and collaborative projects that have evolved from our close proximity. And this is one wonderful example of that, where we've shown that a compound identified in a high-throughput screen to promote cell matrix connectivity is showing these really pronounced effects on accelerating myogenesis. So these are C2C12 cells, 4 days post-differentiation. You can see the vehicle control here, with the red being the myosin heavy chain. And when we add increasing amounts of this compound X, we'll call it, we see the myotube size just increases dramatically. This is quantified here in this, this plot below, showing you the area coverage of these myotubes. So we're seeing significant gains here in myotube size, which is also proportional with protein content. So we think that this compound has really interesting potential to accelerate and make the differentiation process more efficient. And importantly, we're also mapping mechanisms of how this compound is working, which could identify additional targets for future follow-up. On the front of the adipose tissue generation, we've been working to conduct high-throughput screens of natural and food-grade compounds together with our collaborator, Dr. Robert Demoiseau, the molecular shared screening resource at UCLA. And here is just an example of two of the compounds that emerged as top hits in this screen. The rosiglitazone is our positive control here. And you can see both combinations of these compounds and rosiglitazone increase the lipid content, as shown in the green, dramatically compared to our vehicle. And we've done some preliminary synergy tests revealing that in combination, compounds A and B are showing dramatic increases in the lipid accumulation after only 4 days post-differentiation. This you can see by this color map gradient. And what's very interesting is that the 0.1 micromolar rosiglitazone is around this like greenish color. 1 micromolar rosiglitazone is the red color. So these compounds are achieving similar levels of increased lipid accumulation as the rosiglitazone control. So we're very excited to follow up on these studies as well. Again, these were with 3D3L1 adipocytes, so testing in other cell types as well will be an important next step. So finally, we all acknowledge that consumer perceptions and can be a challenge in the field of cultured meat, and to that end, we've worked with our collaborator in behavioral sciences, Janet Tomoyama at UCLA, and I know we'll hear more about this topic later today from one of our illustrious speakers. But I think this just underscores the importance of how research and development can contribute to addressing some of these concerns, and together with development of science communication techniques to engage some of the general audiences in these discussions, I think will be really critical. So we're excited that we have this platform that we've developed through Science and Food at UCLA to extend both into science curriculum for our UCLA students and also K through 12 students, and also this through our public events that are now held in partnership with many different partners across campus. This will be an important vehicle, I think, to be able to bring together chefs and scientists and to in leading this dialogue on these important topics related to cultured meat into the future. This is just our latest event here with Chef Virgilio Martinez that we held recently. So using similar formats with these with chefs is we found to be a very successful way to engage large numbers of public audiences in our conversations. So I'd just like to acknowledge all of the amazing people I've highlighted, Sam and Stephanie, who did much of this work, and also Qingwen as well as our various funding sources and collaborators, and happy to take questions.
Thank you very much. Yeah, I already see a question over there. Is the cube coming, or shall I bring the microphone? Whichever one's first.
Hi, good morning. Thank you for your presentation. Actually, we read your article, the first article that you showed about the microcarriers, the gelatin microcarriers. We read it some months ago, actually in a journal club. And I remember some questions we had back then. I remember you made microgrooves in the gelatin scaffolds.
Yes.
But I remember from the article that actually the muscle differentiation was not better with the grooves.
Right.
So my question now is, do you know why it did not improve the differentiation? Because there have been articles before that where it did.
Right.
And are you still using the grooved material, or you just...
This is a great question. So we were surprised that there were no significant differences between the spherical and the grooved microcarriers. In this context, as I showed, the cells and the microcarriers aggregate together to form these sort of three-dimensional they're three-dimensional spheroids, so the cells are experiencing a quasi-3D environment. So there are other factors that I think are at play in contrast to some of the studies showing that the grooved substrates, that topology can drive in a 2D environment the differentiation. So we think those other cell-cell contexts, for example, other factors could be at play. It's also in suspension culture, so there could be fluid shear stresses that are impacting these behaviors as well. But it's an excellent question, and we don't fully understand the answer yet, but it's an ongoing topic. So then the question is, well, what's the value of the grooves? Is it even worth it to go through the hassle of making those microcarriers? And many of our studies currently are just focusing with the spherical microcarriers, which are very easy and fast to make. But that having been said, I do think there are interesting avenues to explore with the grooved microcarriers. Well, we didn't see dramatic effects on on the myogenesis. Considering the 3D tissue, the aspect ratio of these grooved microcarriers could be really interesting to explore. Having a higher surface area to volume ratio could reduce potentially the overall volume of the microcarriers in the context of the of a 3D tissue. Perhaps they would be remodeled or degraded differently by the cells in this microenvironment as well. So there are some interesting avenues, I think, to really understand how the geometry of the microcarriers might impact the overall texture of the meat, of the tissue. And that's something for future studies as well.
Yeah, it's also something that we have been thinking about, because all the microcarriers now are spherical, but is that the best shape?
Right.
Yeah. Okay, thank you
It's
for
a good
your
question.
answer.
Yeah.
Thanks for the great talk. You did mention that gelatin can be replaced by pectin or alginate. Have you tried that in the lab?
Yes, we've been making both. Well, in fact, the method for making these microcarriers we've used for many years to generate agarose particles as calibration particles for some of our mechanical measurements. For the cultured meat, we've also been making pectin microcarriers as well. So yes, we've been adapting this approach.
And did you have to
materials.
modify it for cell adhesion?
Yes, we're finding that there needs to be some either collagen, as Stephanie showed, integrated into the pectin microcarrier, or functionalized, like RGD functionalized, for example, to promote the adhesion.
Right, thanks.
So if there's time for one more question.
Went surprisingly well. So you see a clear optimum when it comes to the mechanical environment in cultivating these cells on microcarriers. Can you relate that to the natural environment that these cells are growing in? And can you use that information also to optimize surfaces for specific cells in terms of mechanical properties?
Yes, and so I mentioned the optimal sort of stiffnesses that have been found previously for the muscle and the fat, so customizing for the adipocytes with the more compliant matrix stiffness, the Young's modulus of skeletal muscle has been found to be around 12 to 14 kilopascals. So with more fibrotic muscles being much stiffer. So it seems that there could be some physiological basis for this optimal stiffness.
Yeah.
How that translates across species is another interesting question that has been underexplored. A lot of this work is on for biomedical contexts and with mouse,
Yeah.
human cells. So does that answer your question?
Yeah, sure.
Yeah.
Thanks.
Yeah.
So thank you very much. Thanks again,
Thank
Amy.
you.